Alright, Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh, and very good afternoon, everybody. I hope that everybody have had your lunch. Okay, so just to make sure that I'll try and cover everything within this lecture, I'll just start straight away, even if the class is still not full. Okay, so as per last week, if you have downloaded the whole Everywhere app, you can open it now. I do have a few questions, like a tutorial embedded in lecture style. So you can either open your app, or you can go to this wholeev.com and then put in my name as the username. Okay, 019. All right. So first question. I have not activated that one. Okay, so the first question, which one is a beta sheet formation? If you look at the structure, A, B, or C? Okay, so if you want to do it now, you can do it now. Anyone else wants to respond? Okay, I have six responses now. Okay, 7. Eight. And maybe one more, then next one. Okay, so that one is correct. So the answer is C. Okay, so C, if you notice at the back there, so that is the beta sheet. So A is going that way, so it's an alpha helix. Okay, this is based on all the structures are not covered in this lecture, but at least you should already know the basic ones because it was covered under SID3004. And B is the random coil. Okay, all right. So let's move to the next one. So next one, which one of these do you think will bind to the quadrant? Okay, the quadrant meaning this quadrant over here. So which one of these, oops, I shouldn't be writing that way. Okay, so which one do you think will bind to that quadrant? Anyone else is trying? I have 10 now, 11. Okay, anyone else? There's still about 20 seconds left. If you want to try, then you can quickly go into paulf.com and then key in Hadi Fuad 049, okay, as listed there. Okay, 10 more seconds. 9, 8, 7, 6, 5, 4, 3, 2, and 1. Okay, so the answer is all of the above. Okay, because towards the end of the day, the question is asking which one do you think will bind. Okay, I'm not asking which one binds the best. Okay, which one will bind. So the basic theory of, if you recall, in enzyme kinetics, is that you have an active site and then you have a molecule that can bind to the active site. But whether the activity of the enzyme will work or not will depend on whether the molecule fits to the contour properly or not. Okay, so in this case, yes, D will be the best. Okay, but the question asks which one do you think will bind. So A will also bind. It will have a contact on the left hand side and the bottom side of the circle. Okay, B will have a contact on the bottom side of the triangle. C will also have the bottom side which binds to the bottom side of the quadrant, and then D will also have the highest, the best binding ability compared to the other four. But because the question asks which one will bind, then the answer should be all of the above. Okay. All right. So this bit was not covered last week. So what me and my colleagues are trying to do is we are trying to improve our way of doing teaching and learning online, via online, and not this kind of online. It's more on the asynchronous method, whereby we do a recording and then pass it to you guys. Okay. So as you've seen, if we do a live lecture like this, I can have questions put on during the lectures, and then you can participate in the lecture. Now, something which is you will not be able to do is during asynchronous lecture, whereby I definitely cannot turn on the questions for you guys to actually participate in the practice. Okay, so what we are trying to do is we are trying to use a platform in a Spectrum where we can embed. I think some of you guys might have encountered this during your SID3004. Okay, so whereby the question will be embedded in the pre-recorded lectures. Okay, so that's what we are trying to do. If you want to try and participate, you can QR this QR code. Okay, you can scan this QR code quickly, and then just answer like very few questions. Okay, but for today, the type of intervention, if you want to participate, is just A. Okay, A means that it's a live lecture like this, and B is when the lecture is given as a recorded lectures with the question embedded during the lectures. Okay, and help us by participating. Doesn't mean that if you don't want to participate, we do have a lot of. This is just a participant. I just want to see teaching and learning is you guys, whether you guys think it's better or it's better just to give just a blank lecture without any things that you can participate in. Okay, and since it's COVID and we have a grant for this, we are thinking of reimbursing the participants, okay, by giving something which we have not yet decided, because we need to talk to the grant provider whether we can actually do this or not. But that is something that we are planning to do. Okay, so if you want to participate, again, just scan the QR code. Today's attendance is COVID vaccine 2022. Okay, just scan this quickly. Otherwise, by the end of this lecture, there's another QR code where you can scan for your attendance. All right, so let's move on to our lecture for today. So we will look at synthetic and biosynthetic pathway of producing peptides and proteins. In SID2002, you've looked at how do you actually synthesize the amino acids in a very kind of like specific manner. And then if you have set for SID 3020, which is natural products and biotechnological processes, I've also touched a little bit on how do you actually extract out proteins that Am I still here? Can you guys hear me?
Yes, doctor.
I for a few seconds, I think. There is some issue. This might happen a few times. If it did, then I'm hoping that the recording is good enough for you guys to review if you need to. Okay. All right. So as I mentioned in SID 3020, I've did a little bit on how do you actually extract out the proteins that have been synthesized using biotechnological processes, and that is what will be covered a little bit, not in terms of the purification, but in terms of the technology. Okay, so when we move, if you go back to SID 2004, this is what we have learned. We know that there are 20 amino acids that is being recognized by IUPAC system as the natural amino acids. Okay, meaning that there are a lot of amino acids in the world. There are 20 critical amino acids that builds us human as what we are. So but if you consider bacteria and other organisms, there are actually more than 20. Okay, but the IUPAC system recognizes as the amino acid for now. Okay, in the future or the year after, they might change it, but as of today, there are still 20 amino acids that are being recognized as the natural amino acids. All right, and we also looked at the multi-step processes of citric acid cycle, whereby our body managed to produce certain amino acids, while the other amino acids we need to take it from by ingestion. Okay, so if you recall, we have two categories of amino acid. One is essential, the other one is non-essential. Okay, so the difference is essential amino acids are the amino acids that you can only have by consuming foods. Okay, non-essential meaning that you what are the essential amino acids, you can go either visit IUPAC website amino acids. Okay, of course, the various produce it's chemically I'm not moving. Am I still here? Quiz. Quiz for today. Let's see. All right. So which of these is not the synthetic approach to synthesize an amino acid? Okay, so this is basically a pop quiz based on SID 2004. So which of these is not a synthetic approach to synthesize an amino acid? Okay, so you have Hell, Bowl, Hart, Zelinsky, reductive amination, Gabriel malonic ester, Striker synthesis, and Xavier Click synthesis. So which one do you think is not a synthetic approach to synthesize an amino acid? Okay, so I have 4. If anyone else wants to join in and have a try. I have 5 now, 6. So 15 more seconds. 50-50. Xavier Click is going higher. Striker synthesis. Okay, reductive amination. All right. Thank you for participating. The answer is Xavier Click. Okay, so Xavier Click is I'm not sure if it actually exists. That is a name of a synthesis that I created when I created this question. Xavier means it's Professor Xavier, so X-Men. Okay, and Click I just put it in so that it sounded like a chemistry thingy. Okay, but Click is not a synthetic approach. Okay, so HBZ is correct. Okay, HBZ is okay. This is the correct one. Reductive amination is also correct. Okay, Gabriel malonic ester is also a correct way, and Strecker synthesis is also a good one. Okay, so Click is a made-up synthesis that I've created. Hopefully, it's actually not true. Okay, thank you. Now, again, this is a little bit of a refresher. So in any synthetic approach, okay, regardless whether it's peptides or proteins or any reactions that you have completed for the past, during your first year, especially because your second year is most likely just an online version. Okay, so any synthesis reaction that you have completed during your first year at least is basically a combination of two products or two reactants. So you have an A, you have a B, you combine them, stir it for a while. For B, you might add a few, and then after a while, a white powder forms, or this, when you crystallize it, it forms a powder. Okay, so that is a basic synthetic approach for everything in chemistry. So is there any difference compared to a peptide or protein synthesis? Well, hopefully your answer is right, which is no. Okay, so there is no difference for a peptide or protein synthesis if you're thinking about chemical synthesis. Okay, if you're just focusing on the chemical synthesis, so basically if you have one amino acid, in this case a glycine, and you have an alanine, a second amino acid, you have the reagents or the catalyst that is required for the synthesis. You put it in a solvent, for example, dimethyl formamide, okay, or we call it as DMF. You put it in, you stir it, leave it for between 30 minutes to 2 hours, and then you will have your diamino acid. Okay, bit ugly drawing, but should serve the purpose. Okay, you have a diamino acid. Wait, I think I missed one. All right, so that is the basic concept. But of course, in this case, they can be a lot of mixture. So there's a lot of diastereomer. Okay, so that one is one diastereomer whereby you have a G and an A, like that. Okay, you can also have a G and a G, a G, a and an A, and then finally you can have an A and a G. Okay, so if you have these two amino acids and then you mix them, there are 4 outcomes. This is a refresher for SID3019. And then to make sure that this does not happen, you introduce something that you call as protecting group. Okay, a refresher. I'm not going to go into detail about protecting group and whatnot. This is just a refresher. Okay, so to focus about the synthesis in more detail, okay, you have, if you're thinking about the industrial perspective, for example, so there are 2 different options that they can proceed. One is the synthetic approach, whereby they can either use an automatic system, okay, or a manual, whereby you yourself or a person in the company actually mix one amino acid to another and then do the same process, repeat the process until you manage to synthesize the whole proteins or peptides. Okay. Or the industry can also proceed using a biosynthetic approach, which in this case, it's related to the subject SID3020, biotechnological processes. Okay, but over here, we are trying to, if you have set for that particular course, I actually did not go into very detail on how you actually do it. So this course, I'm hoping that to give a little bit more detail. so that you can actually see when we are talking about all the examples about protein and peptides technology, these are some of the approaches that the company can take or industry can take to actually produce a peptide or protein that can be used for a different technology. Okay, so as I mentioned previously, the technology that we are going to look at is one of them is, for example, COVID-19 rapid kit. Okay, the rapid kit, if you have done one, okay, so it's a small kit whereby you just put a drop of your blood, and then you put in some solvents, a drop or two of solvent, and then you will just carry your blood or your sera actually, and then if there is a COVID-19 antibodies already in your body, then you get a signal. Okay? If there's none, then you don't get the signal. So that technology is actually based on protein and peptides. Okay, so it's an applied technology. By knowing how to actually use protein and peptides, you can actually produce this type of a technology. Okay, so moving back to the lecture notes. Okay, so the companies in the industry can have two different options. One is synthetic, one is biosynthetic. But if you're looking, if you are interested in looking at the synthetic approach, there are additionally two different branches. So one is a solution phase. Okay, solution phase synthesis. The other one is a solid phase synthesis. Okay, so we will go into a little bit more detail on the solid and solution phase, what are the difference, and so on and so forth. And if the industry is interested in using the biosynthetic approach, there are more than three, obviously, but only these three are being the major focus for this particular lecture. Okay, the first one is a cell-based synthesis. So the cell-based synthesis is more or less related to the ones I've already covered in SID3020. Okay, so we're going to look at the technology in kind of like a brief. Second one is a cell-free synthesis, meaning that you actually do not need to use a cell per se. Okay, so you do not need to use a living cell to actually produce your proteins or peptides. And the second one is you can actually use a plant. Okay, so it's a plant-based synthesis. So we as a human, we also produce proteins and peptides. Animals also produce protein and peptides, and we can use them. Okay, but in ethical perspective, to use an animal or human to actually produce something that is going to be used by humans are really not ethical. Okay, so we use something which cannot feel pain. We are hoping that plants do not feel pain. Okay, so that's why plant is part of a biosynthetic approach to produce any proteins or peptides of interest. Okay, and of course, if you're talking about producing proteins and peptides, you need to know the relationship. Peptides. Okay, so you need to know the relationship between this. So what is gene? What is DNA? So you know that in our body, in our cell, we do have our own DNA. Okay, and that DNA we call it as gene. So gene is a sequence of DNA that produces something. Okay, so that is one jargon for today. And then you need to know what is cDNA and mRNA. So I'm sure at least you guys have heard, if you have never encountered any biology, at least you have heard about mRNA. Okay, so mRNA is what Pfizer is using for the COVID-19 vaccine. So it's mRNA. Okay, so that's another jargon. And then from there, you need to know antibody. So antibody is a type of a protein. Okay, but it's a more specific protein, and we will cover about antibodies, I think, in the next lecture, next week. Okay, and all of these are interrelated to one another. So from gene, you can produce a protein. From a protein, you can know the cDNA or mRNA. And while knowing the mRNA, you can also produce antibody. Okay, so everything are interconnected. So there's no standalone DNA and then doesn't produce anything, or mRNA that doesn't produce anything. Okay, so these are all interrelated, and these are what the industry uses to produce our COVID-19 vaccine, to produce our COVID-19 test kit. If you are getting married or if you are going to, okay, so you will need to do HIV tests. Okay, so HIV test is also using the same concept that we are going to touch. If you are, well, basically any test kit, okay, a rapid test kit that you might have accounted in the future. For example, glucose level reading. Glucose is different. No, sorry. So there's a lot of disease monitoring, for example. So a lot of it is actually a technology based on antibody. Okay, so we will look at antibody in a bit more detail next week. So for today, we'll just focus on these 5 ways where you can actually produce peptides and proteins. All right, so if you're focusing on the synthetic pathway, as I mentioned, there are 2 pathways. One is solution phase, the other one is a solid phase. So a solution phase is basically the same way whereby you are doing, whereby you did your synthesis during your first year. So basically you have A, you have B, you mix them together, put in some other stuff, and then voila, you magically manage to produce a product. Okay, so it's the same way. So if you are to proceed with a solution phase, then you need to have A, an amino acid, plus a B amino acid. It's the same way as I mentioned earlier. You have some reagent or catalyst, and then you will have your A plus B amino acid or dipeptide. Okay, a solid phase, on the other hand, is like a surface chemistry but cooler, in the sense that instead of just mixing, again, as I mentioned, if you have an A and B like this, if you mix them together, you can have 4 different diastromers. Okay, but if you have a solid phase, what it means is that during your synthesis, the first step that you will have is you have a solid support, whereby the solid support has a functional group. Okay, in this case, the ones that I'm showing here as an example is you have an amine group. Okay, and then what you do first is, if say the sequence that you want is A and then B. Okay, so the sequence is A, B. For example, if you want to have more A, B, and A, B, for example. So what you do is you start off with just giving a B over here, because remember, an amino acid has 2 function groups like that. Okay, so if you have a B, this is a simplified version. Okay, this is not what people do. This is just a simplified version. So what you do is, instead of mixing A and B together, whereby you can get a lot more diastromer, what you do is you have this solid support, and then you put in only B inside there, so that the first reaction that you will get is only B. Of course, you can have B, multiples of B's and whatnot, but just for simplicity, you only will have B. And then once you have your solid support, you can add in A, so you get a connection with an A, and then you can add B again, and then A again, and so on and so forth. So it's kind of like growing a peptide 1 amino acid at a time, but by doing this, you can actually control the synthesis properly. Then towards the end of the day, whatever you want to synthesize, that is what you will get. All right, and so the minor differences between these two are the protecting groups. So for a solution phase, normally the amino acid is using a different protecting group, and for a solid phase, you are using a different protecting group. Why? Because for a solution phase, nowadays everybody wants to talk about green chemistry, if you know, or sustainability of something. So in chemistry, part of the sustainable concept is sustainability in terms of synthesizing or doing a chemical reaction. Okay, so in this case, the solution phase reaction can actually be done in just water. Okay, so if you are doing a synthesis in water, meaning that you can get water everywhere around the world. It's very cheap. It's very easy to clean it. Okay, so it's part of a sustainability. So in order for you to be able to synthesize an amino acid with a protecting group in water, the protecting group cannot be the same as the one that you are synthesizing in a solid support, whereby the solid support is normally done in an organic solvent, such as DMF. Okay, so DMF is less sustainable, it's toxic, and so on and so forth. Now do a synthesis of in water. Of course, water is very hydrophilic. DMF depends. Some solvent, organic solvent, not DMF per se, some organic solvents are not hydrophilic, and therefore, to make sure that everything goes smoothly, you need to change the protecting group from the commonly compatible, common protecting group that are compatible with an organic solvent. You need to have different ones that are more compatible to an aqueous environment. Okay, so that's why I said the minor differences is in the protecting group. That's one. Second one is the reagent. Okay, the current reagent, so the technology that first developed was the first developed is of course the solution phase, but then the technology that progresses a lot are actually the solid phase. Okay, so the technology in terms of synthesizing peptides and proteins are advancing rapidly in an organic solvent, so a more pure chemistry type of reaction. So the reagent that was produced initially are compatible only in organic solvent, and they are not compatible in aqueous, because as you probably notice, you have a lot of functional groups that are very, very easily hydrolyzed in water. Okay, so that's why the reagents needs to be changed. So if you have a reagent that can be used in a solid phase, you cannot use the same reagent for a solution phase synthesis. Okay. Other than that, of course, the coupling meaning that when you want to add one amino acid to another, so that's what we call it as coupling. So you couple one amino acid to in a stepwise manner. Okay, deprotection meaning you want to remove the protecting group. Again, if you don't recall, go back to SID3019. This is actually covered there. And then the cleavage of the peptides from the solid support, so that finally you can get your own protein or peptides. Okay, but in terms of the chemistry, the concept about connecting the carboxylic acid group to a free amine is the same. Okay, it's the same for both solution phase and solid phase. So chemistry are almost similar, but all the reagents and the way you conduct the reaction is different. Okay, to get into a little bit more detail, so this is the a protocol that I've taken out from a research manuscript. So this is just to show you in the solution phase how they actually do it. So you have a different, in this case, a phenylalanine with O-methyl group with HCl salt. So you know when it's a salt, it's very compatible in water. Okay, so you have that. Initially you put it in chloroform. You have N-methyl methacrylate. Okay, you add it, you mix it, and so some things that I want to let you focus on is one is time. Okay. Second one is scale of the synthesis. The third is the solvent that is being used, and then number four is the processes. Okay, you can go back after this and look into the fine detail, but just to go about in a very quick and rough manner, so you can see. Probably can add something else, temperature, because that one is missing. Okay, so temperature over here, they do it in 0 degrees. So again, 0 degrees. You can see if you're talking about time-wise, the reaction needs to go on for about 24 to 36 hours. Okay, solvents that they use is different. You will see, and the process, if you read through all the methodology, the steps will be different. Okay, so this is one example of a solution phase. Now we will look at how you actually do it in a solid phase. I just want to change my slide. Okay, so this one is a solid phase. Again, if you focus on time, scale, solvent, and processes, then you should be able to notice that it's totally different. Even though the chemistry is similar, but it's different. So for one thing, you can look at 2 minutes. Okay, so 2 minutes is for activation of amino acid, and then for coupling, it's just 20 minutes. Compared to previously, it's 24 to 36 hours. So these are some of the pro and cons of doing a chemical synthesis of peptides and protein. If you were to compare between a solution synthesis approach and a solid phase approach. Okay, and well, you can actually read it at your own time and actually see on the differences. The critical bit for this section is to actually know that these 4 keywords. Okay, so you need to understand what are the differences, but it doesn't mean that you need to memorize everything. You do need to memorize this and that. What you need to know, what you need to understand is there are differences between a solid phase synthesis and a solution phase synthesis. Okay, and of course, one of the processes that are different between a solution phase and a solid phase is that the resin, okay, this resin up here, so this is the solid support, the black dot that I've drawn in the previous slide. Okay, you just need to wash it multiple times between each step to remove all unwanted reaction, compared to a solution phase whereby you need to do, what did it say? Okay, so you need to do extraction, and then organic layer was dried over anhydrous potassium sulfate, and then you filter it, you evaporate it, and so on and so forth. So the processes are more complex in a solution phase, even though it is more sustainable. Okay. All right, so you can just complete this on your own. Read through the 2 paragraphs from the 2 previous slides, and then you can just fill this up. Okay, I want to skip through because of time limitation. All right, so in summary, the major differences are the medium, solvents that is being used. Okay, so the reagents, if you go back and see, one is using M&M and chloroform, the other one is using DMF, and of course you can actually use water. So the example that I've shown just now was not water, it's chloroform, but you can actually use water. Okay, and of course the protecting group. Okay, so but the flow of the synthesis over the right hand side is similar. So either you have a resin or not, it depends on solution or solid phase. But once you have your amino acid, the first one is you activate it to make sure that the reaction can be proceed very, very quick or as quick as possible. And then you coupling steps whereby you couple the 2 amino acids together, and then either you cleave to get the peptides or proteins, or you continue with the deprotection of the protecting group, and then again activation and then coupling, so you can actually grow your peptide. Okay, so these are the general concept for a synthetic approach of peptides and proteins. Okay, now let's move on to the second one, which is a biosynthetic. So as I mentioned, there are 3 different approaches. One is a cell-based synthesis. Let me just, okay. One is a cell-based synthesis. The second one is a cell-free synthesis, and the third one is a plant-based synthesis. Okay, so that is one of the things that is actually growing. So the industry is more nowadays. It's more interested in doing a plant-based synthesis. Why? Because one, to use a cell-based, it's more costly and it's time-consuming. So imagine that you can ask a plant, okay, a tree outside of your house, to grow a peptide that you want. Okay, so it will grow unlimited. So there will be unlimited supply. You just need to put probably some soil, water it once in a while, and then we can just pluck on the leaves and extract out your peptides or proteins. Okay, so that's why the technology is now shifting from a cell-based to a cell-free. and then now more focus is being given to a plant-based biosynthesis. All right, so we looked at the differences between cell-based, cell-free, and plant synthesis. So the basic things that are difference between those three are: one is the host, okay. Second is the method whereby you introduce the technology, and the third one is post biosynthesis. So after you get the protein, okay, from say cell or cell-free or plant options, what are the next step? Okay, and all these three categories belong to one terminology, which is recombinant protein expression technology. Okay, so this is what it's a very broad, it's a very huge technology, and this technology can be used in multiple areas, and the three areas that I'm going to touch is cell, cell-free, and plant-based. Okay. All right. So let's focus on the first one, which is a cell-based production. Okay. So you can produce a peptide and a protein inside the cell. So this is the same as what was covered in SID3020. Again, I'm not sure whether you guys have sat for that particular course. But if you have not, then it's basically talking about that. Okay, in a very brief. If you have, then I don't need to talk a little bit in more detail. Right. So for a cell base, there are still it's still a huge consideration. So there are still huge consideration, and one of them are the host selection. Okay, when we're talking about cell, it can be a human cell, it can be a bacterial cell, it can be a yeast cell, it can be eukaryote cells, it can be plant cell. Even plant cells can be categorized in here. Okay, or insect cells. So there are multiple options that you can actually choose from to actually produce peptides or proteins of interest. The difference is that one is bacteria. Okay, the difference are actually, I don't want to go into very detail, but because of these cells are unique to that particular organism, what we call as post translational modification are either absent or present in some cells. Okay, for example, if you were to synthesize an insulin in a mammalian cells, okay, basically when we are talking about protein synthesis, we always talk about a pure protein synthesis, meaning that if you produce something like that, it will just be a protein. But in reality, when we are talking about synthesis of a product, okay, it's not always a stick and stone where when you're talking about proteins, it will just be protein. So a protein in a biosynthesis can be either glycosylated, phosphorylated, and so on and so forth. So there's a lot of call it post-translation that can be done of what are the post-translational modification that can occur. So you have a few options. Okay, yeast, E. coli, those two are not together, even though they are both microbials, but they don't belong in the same category. So one is a bacteria, one is a eukaryote. Okay, insects. So insects are insects, so they don't belong to bacteria or microorganism, and also they don't belong to a mammalian like us. Okay, and of course you can have a transgenic plant. So we will look at a plant base later on in the slide. Oh, I only have 5 minutes. All right, so basically that is the general technology for all types of biosynthetic process for proteins and peptides. So they are more or less the same. The path is, of course, you need to transform, meaning that you have a gene of interest, your DNA of interest or mRNA of interest that you want, then you will put it into an organism. So in this case, the example is a bacteria, okay? But if you are to talk about a plant biosynthesis, then the organism of interest is the plant itself. So you either put it in bacteria or you put it in a plant. But for a cell-free synthesis, you actually skip that particular process. So you don't put it in any organism, okay? And you put it into something that we call as vector. And this vector, when you put it into bacteria, so once there is a trigger, then the bacteria or the host, okay, will produce the protein of interest. Okay, so of course this is just one way to look at it, but you normally, once you have one single cell that have the protein that you want, you will first grow the cells into thousands or millions, and then you put in a trigger so that each cell will produce the protein or peptides of interest. Okay, so that's one way to do it, and we will look at this pathway in more detail when we are talking about vaccine technology or while we are producing more proteins. Okay, so this is the general idea on how you can produce proteins and peptides using a biotechnological approaches, or we can call it as a biosynthetic approach. Okay, so either or is fine. All right, 4 more minutes. Okay, so this is, as I mentioned just now, wide cell-based or wide range of host selection. So different hosts has different results, glycosylation, phosphorylation, etc. Expression of a single chemically synthesized cDNA coding for human pro-insulin, followed by purification and subsequent excision of the C peptide by proteolytic digestion. So this is a sentence. So this is the original technology that was used by Eli Lilly way back in 1982 to produce insulin for diabetic patient. Okay, so this is already an industrial application. So it's not just about theory, it's not just about learning about it. This is actually what the industry is using. So instead of opting for a total synthesis, sometimes it's cheaper to produce in a biosynthetic approach. Okay, all right. So this is a cell-free based synthesis. So in vivo system is, for example, is a cell-based that we just talked about in the previous slide, and CFPS is the cell-free system, whereby you can see here the steps are slightly different. Okay, but the most critical thing, the similarity between those two technology is this. So one, you have the gene or the DNA of that particular protein. Over here, you have the DNA template. Okay, so those are the two different things. Otherwise, everything else is more or less the same. You need to grow cell. Over here, you also grow a cell. Okay, but in the cell-free system, you kill the cells and collect everything else from the cell. Over here, you kill the cell at the very end to extract out your protein. So the technology are more or less the same, but it's different in the sense that which step the DNA of or mRNA of the protein is being introduced to the system. But towards the end of the day, you still grow cells in both techniques, technology, and then towards the end of the day, you will have your own protein. Okay. All right. So you can have a look at this particular article. It talks about a little bit more on cell-free system. If you are interested, then you can just go in, and this is just an extract from that particular paper. It's more or less the same as what is being shown in the previous slide. So first you grow the cell, and then you extract, or you kill the cells or lyse the cell and extract out all the cell component. You can either lyophilize and store it, and then you rehydrate before you want to use it. So this is one advantage of using a cell-free base synthesis compared to a cell-based. Because a cell-based cell is a living cell, so you need to keep on feeding it and whatnot. So to make sure that it's still alive. But a cell-free synthesis, you can actually grow the cell, kill it, store it, and then if you want to reuse it, you can just extract out, rehydrate, and then use it. Okay, so those are the two major differences. And the third one is a plant-based biosynthesis, whereby you still have your vector, okay, and you still have your bacteria. But in this case, it's not a bacteria, it's agrobacterium, it's a specific bacteria that can infect a plant. And then once it's infected, this plant can actually produce a protein. Okay, that's in this very quick summary. Everything else you can just read through. It's very simple. So this is a summary of the biosynthesis approach. You either have your DNA, first off, you need to have your DNA of interest, regardless of what approach you want. You need to have your DNA of interest that has the sequence of the protein or peptides of interest. And then you put in, either you put in now or later into the cells. And then finally, the organism or the host will produce the protein of interest. You purify it, then you'll get the protein. Okay, so that is the basic concept. We'll cover this example in next week. Okay, again, this is the attendance for today. You can quickly scan it, and that's it from me for today. If you do have any questions, clarification that you want me to clarify in the next coming lecture, just text in the WhatsApp group. Okay, and we'll answer